Today we'll see the topic bacterial growth curve. So bacteria usually grows just by doing a binary fission. There are different types of growth curves like growth cycle, biphasic growth, maintenance of cells in exponential phase, synchronous growth, and bacterial growth in vivo. So now we'll see the growth cycle, how the bacteria is dividing and how the bacterial number it is increasing. Now we'll see the bacterial growth. So generally this bacterial growth is regulated by nutritional environment. If the nutrition is good, if the nutrients are supplied very well, then automatically the bacteria undergoes the cell division process. So that is a better environment which make a bacteria to divide. So that means environmental factors, for example nutrients and pH levels, temperature, or deciding the bacterial growth. So this bacterial growth can be plotted in a graphical way called as a growth curve. So the graphical representation of the bacterial growth is called as bacterial growth curve. It is having different phases, different segments like lag phase, log phase, or exponential phase, and third one stationary phase, and next one decline phase. So this is the graphical representation of the bacterial growth cycle. So the bacterial cell division, the bacterial growth can be plotted like this. So here you can see different phases, and they were given in different colors too. So the first one is the lag phase and followed by log phase, and there is a constant period called a stationary phase followed by death phase or decline phase. So if you have plotted the time on x-axis and the number of bacteria on y-axis, where you can see this lag phase, log phase, stationary phase, and the death phase. So these are the four phases of the bacterial growth curve. Now we'll see one by one what is this lag phase. So generally bacteria can able to divide just within 24 hours. Just let us assume that we would have provided the required nutrients for the bacteria, and we have inoculated a few cells into that medium. So at the time the bacteria are in lag phase. Generally it lasts about 1 to 4 hours. So in this lag phase what happens? So as we have supplied the nutrients, we have given a new environment for the bacteria. In that case what happens means that the bacteria used to adopt itself to the conditions what we have provided. In this phase what happens means the synthesis of the RNA takes place, and other enzymes and other molecules which are required for the division of the cells. So this takes place during this lag phase. Next, followed by log phase. So in lag phase we couldn't see any kind of cell division. Because of that only here you can see it is a straight line in lag phase. So here you can see in lag phase the number of cells are going to be constant. We couldn't expect the increase in cell number. So this is a lag phase. And whereas in log phase, this log phase is also called as exponential phase or logarithmic phase. So why this is called as exponential phase means the doubling of the bacterial cells takes place by following the formula 2 to the power of n. So in generally 2 into 2 into 2 into 2 like that, the 2 to the power of n formula it is following at the time of doubling of the cells, cell growth of the bacteria. So with this what happens, automatically the doubling of the bacterial cells takes place drastically. The doubling of the cells takes place very drastically. So as there is a more and more number of of bacterial cells growth is taking place, this is called a log phase or exponential phase. So this may last around 8 hours. This phase maybe lasts if we don't provide the fresh nutrients. Okay, so in log phase what happens means that the cell number it is increasing by following the formula 2 to the power of n. Next, so this number continues till the nutrients are going to get depleted. Next, the third phase is a stationary phase. So stationary, constant. So this is due to the stationary phase is due to growth limiting factors are going to be depleted. So initially I have told you that we are going to provide the nutrients. So we are providing the nutrients means automatically the bacteria they are going to utilize those nutrients, and they are involved in the growth process, the cell division process. But what happens means as the bacteria is getting nutrients from the medium, and those nutrients are utilized by the bacteria, and this bacteria undergoes the exponential way of cell division, and the number of the bacteria are going to increase drastically, which takes place in exponential phase. What happens means later on this bacteria which are there in the medium, as the number of bacteria is increased, the nutrients automatically they are going to be depleted. So with this what happens, the older cells may die, and the new cells may form. So this what happens, the number of new cells were formed may be equal to the number of cells dead. So with this what happens, we couldn't find the cell number increase during this stationary phase, so that we used to maintain this straight line. We can see a straight line that indicates that the number of cells dying is equal to number of cells the number of new cells forming. So with this what happens, we couldn't find the increase in cell number. So because of that only in graph we used to see a smooth straight line. So this may last about a few hours. Next, the next phase is a decline phase. So with this what happens, as we are not supplying the fresh nutrients, the more nutrients to the bacteria, with this what happens, automatically the nutrients are going to be depleted very drastically, and the older cells were already died, and there is no chance of new cells to form. With this what happens, automatically we can see the downfall in bacterial cell number. So this decline phase indicates the downfall in bacterial cells number. So this is due to the completion of nutrients, the extraction of the nutrients, and as the cells are metabolizing, there is every possibility of release of some metabolites which may become toxic to the other living cells. So with this what happens, accumulation of the toxic products and autolytic enzymes. So with this accumulation of such a kind of metabolites may also make that bacteria to die. So with this what happens, there is a downfall, there is a decrease, there is a lowering of the bacterial cells takes place. So this is a decline phase. Next, there is one word which is related to bacterial growth curve is synchronous growth. So generally bacteria used to grow very non-synchronously in ordinary culture medium. So that means the bacteria, based on its metabolites and its activity rate, the bacterial cells may not go for synchronous growth. But we can see the bacteria which are there in culture medium, where simultaneously show different levels of growth called synchronous growth. So that means if there were 100 bacterial cells in the culture medium, means if the all the 100 cells are going for cell division at the same time, means we can see all the cells were in log phase. And if the cells are maintaining, all the cells are in what we call stationary phase, means all the cells we can say that they are in synchronous growth. Next up, bacterial growth in vivo. So in vivo means in natural conditions. So the bacteria which are there in our body grows very slowly when compared to the bacteria which are growing in artificial conditions or in laboratory conditions. So that means the bacteria which are growing in laboratory at much faster rate than the bacteria which are growing in our body. So then what are the reasons why this slower growth rate of the bacteria in human bodies or in any other organisms? So this is due to nutritional status of the body and a defense mechanism. Of course, all the organisms possess a kind of defense mechanism which may inhibit the growth of the bacterial cells. So this is the reason why the slower growth rate in vivo conditions compared to in vitro conditions. In vitro conditions means it is nothing but laboratory conditions.